The topic of today is to talk about protein sources, and not only from a nutritional perspective, but I would try to bring in also some other characteristics of protein sources which could be helpful to improve our diet, especially when it comes to intestinal health and, let's say, improvement of the functioning of the immune system. This morning we heard about the growth of the world population and the need for nutrients, not only to, let's say, to feed mankind, but also to feed our animals, because there is an increase in demand for animal protein sources, as we heard. So we're not only talking about the tension, which is when we talk about the relationship between requirements for, on the one hand, feeding animals, feeding mankind, and we also know there is competition when we talk about energy sources when it comes to energy supply. I will start off with a few additional comments about availability of protein sources, but the remainder of my talk I would like to focus on nutritional and functional properties of protein sources, and especially how to relate them to improve gut and animal health, and I will end with some conclusions. It's important to state that the number of feed ingredients and protein sources we are using throughout the world is not only about soybean meal and corn. Especially in Western Europe, we are using a lot of byproducts and other protein sources which contribute largely to the protein provision of our animal feeds. So we have, for example, the byproducts of the oil seed industry, not only soybeans, but also rapeseed meal, sunflower meal. We have grain legumes, a number of them indicated here, but we also have byproducts of the starch and ethanol industry. And we have a number of new protein sources of different origin, and already insects were mentioned this morning. The production of insect protein is still in its infancy, but in the long run, we see also options there, and there are a number more. So we talk about a great variety of protein sources which are being used and in the future will be used to a larger extent. So up to now, basically in our animal feeds, we have considered proteins in these ingredients as a source of amino acids and as a source of amino acids for many protein synthesis in the body. However, right now, we are considering other aspects of our ingredients, and for a longer period of time, we considered protein sources also from a negative perspective. For example, we considered anti-nutritional factors being present in a number of legume seeds. So that was a reason to, let's say, put maximum constraints on the use of particular ingredients. And so that was a kind of negative functional value. But right now, we are considering also protein sources also for their ability to release, for example, peptides during the process of digestion in the GI tract, and also as a source of specific amino acids which could have specific effects in body and in metabolism. And then we often talk about, we use the term nutraceuticals. This is coming from human nutrition, where they are a step ahead of us, I can say, because a lot of information we know about nutraceuticals is coming from the human nutrition domain. When we talk about the nutritional value of protein sources, of course we consider the digestive process and of course the release of amino acid in the digestive process. But when we have to think about functionalities of protein sources, we have to also consider the process of protein digestion in terms of release of bioactive proteins and peptides during this process of protein digestion. And that's a new dimension, I would say, to thinking about using protein sources as functional ingredients. So from a classical point of view, we consider ingredients, protein-rich ingredients, as a supplier of free amino acids, and for that we consider mostly ileal digestible amino acids in these protein sources. And we try to match that with the known amino acid requirements for target categories of animals. And more, we also like to consider sources of variation, causes of variation in these amino acid requirements by taking into account effects of genotypes or environment or health status, as we just heard. And a new dimension could be in the future to consider other characteristics of and functions of amino acid in amino acid metabolism, and also consider other functional properties. One of the things which is let's say a plus to the nutritional value is to consider not only the ability of protein sources to release amino acid for metabolism, but also to consider their rate of digestion and release in the GI tract. So we are running a project in which we consider in vitro and in vivo protein digestion kinetics of protein sources. And this is an example of some in vitro work we did quite recently, in which we used a modified Bayesian procedure to mimic in vivo protein digestion in a two-step concept using a pepsin incubation phase and a pancreatine incubation phase. And you can see that for 3 different protein sources, soybean meal, wheat gluten, and rapeseed meal, the kinetics of in vitro digestion is rather different when you compare the different phases of digestion. Of course, we did not only consider in vitro digestion, we also considered in vivo digestion. And here is an example. We are running right now also trials with pigs, but this is an example of the establishment of the kinetics of protein digestion in broilers. For, again, soybean meal, rapeseed meal, and in this case, plasma protein. And when you consider digestibility in different segments of the GI tract and combine that with retention times of digesta in the different compartments, then you can make graphs relating time of digestion in the GI tract with apparent digestibility of protein from these protein sources. And you can see basically that these curves are different in a way, and you can describe that by modeling these graphs. And you see that maybe the maximum capacity is not really different, but you can clearly see that the kinetics characteristics are different among protein sources. That means this will influence not only the location where amino acids are released for use in metabolism, it could also interfere with the release of bioactive proteins and peptides, and that's important to realize. That proteins have certain functional, either desired or undesired characteristics, we know already from the fact that when we consider dietary protein concentrations with the occurrence of, for example, post-weaning diarrhea, that there is an interaction with, between the two. Because generally, if we feed high protein diets to young piglets, that we observe more post-weaning diarrhea, and this effect is enlarged if there is a stimulus in the environment, or if you impose, for example, an eating challenge on these piglets. Likely, or we assume that this is related to more excessive protein fermentation in the gut when you feed diets with increased concentrations of protein, resulting in the release of ammonia, biogenic amides, which could be detrimental to the intestinal lining and to the barrier function of the gut. We know more about functionalities of specific protein sources, and plasma proteins is just an example of a protein source of which we assume that it has certain characteristics beyond the pure nutritional ones. Because we see when we feed to post-weaning piglets diets with plasma proteins, we see a lower occurrence of post-weaning diarrhea, or we see lower fecal scores. And in addition to that, quite some studies have been doing also on other aspects of barrier function, for example. And we see improvement of barrier function if we feed increasing amounts of plasma proteins. So this is an example on how a specific protein source could help to support gut health. Likely this is related to the presence of certain bioactive peptides or constituents in plasma proteins, which could be could have a positive effect on some of the functions of the gut. Well, this is a general overview to help you to realize that amino acids in the diets coming from protein sources do not have a function only as building stone for protein synthesis in the body. We are more and more aware of many other functions individual amino acids do have when they are present in the diet and when they finally appear either in the gut or in the systemic metabolism. And this is just an overview, and protein synthesis is of course in the top, but you can see here a number of effects, individual amino acids, and you have to be of course very specific in that respect, but this is a general overview that it could have effects on, for example, stress responses, on metabolic regulations, on immunity and health, on antioxidative stress and defense capacities of the animals. So one of the things we also know already for some time that, for example, glutamine is a preferred nutritional energetic substrate for the enterocytes in the gut. So if you feed diets, and this is an example with increased concentration of glutamine in the diet, it was shown in post-weaning piglets that the morphology of the gut was improved, increased villus height compared to control diets. And this most likely is related to the fact that glutamine is used as a preferred substrate for energy metabolism of these enterocytes. And especially under conditions of low feed intake, then the amount of glutamine could be relatively low for this particular purpose. So if we thinking one step further, then we have to realize that, let's say, protein sources really could be a source of bioactive peptides. And this is just a revised diagram coming from a paper from the human domain. But if you think that also dietary protein sources for animals could potentially provide antioxidant capacity or immunomodulatory properties or antimicrobial activities, then we could consider potentially also protein sources really as functional ingredients, although we are not that far in establishing these. Here an overview of a paper, again from the human literature. There is a lot of information here, but it's just to illustrate that already there are a large number of proteins elucidated in different ingredients, going from legume seeds to cereal grains, but also to, like you see here, mushrooms, green algae, and so on, having specific biological effects, being either, for example, antibacterial, or being having effects on inflammation, or having effects on, you see here a lot of information on effects of ACE inhibition, and that's related to blood pressure. That's not, let's say, of particular interest, we think, to apply in animal production, but this is related to the fact that all this research is done in the context of human health. But one of the conclusions is also here that many proteins do have a role which goes far beyond the one of providing essential amino acids. And this is the message here, and this is we should take up also in animal nutrition. So in that respect, we can think of also affecting functions of the gut, not only from the perspective of nutrient digestion. Of course, we have to realize what I just mentioned, that some bioactive proteins can arise during the process of digestion in the intestinal lumen. But we could use also this potentially, or maybe via influencing microbiota composition and activity, which relates also to immune system programming. It's already been said already today that major part, or a very important part of the immune system, is located in the gut. So really, programming of the immune system, the local immune system, starts at very early age and could have long-term effects on resistance and resilience of the animal. So here you see the complex interactions in the gut lumen, where you have intestinal microbiota at different locations, and they interact with the intestinal lining, with the enterocytes via all kinds of receptors, and inducing all kinds of signals also to the immune cells which are present in the submucosa. Well, we are only more or less at the beginning to understand all these complex interactions in the intestinal lumen, but we know that dietary composition and substrate present here in the lumen do interfere with these complex interactions. So we have to realize that in the gut, not only carbohydrates, and we know that for a longer period of time, but also potentially proteins could serve as a substrate for fermentation. And we could consider that from a negative perspective, and we have done that for some time already, but we could also consider that from a positive perspective, because it could induce, let's say, on the one hand, what I just said, the release of bioactive proteins and peptides, but also it could result in some compounds which can be on the one hand considered from a negative perspective, but maybe at lower concentrations they might also have positive effects. And we do have indications, and this is an example of a study we did on mice ongoing. We are performing experiments with broilers and with pigs to see what is the effect of feeding different protein sources upon the development of intestinal microbiota in different segments of the GI tract. This is an example using different protein sources from soybean meal to whey protein, wheat gluten meal casein, yellow mealworms, and plasma proteins. And you can see here how microbiota composition was affected in the ileum of mice. We see clear differences, different profiles affected and induced by the use of different protein sources. And we go one step further because we also evaluate the gene expression at mucosal level. And we try to see difference, and then we find difference also in gene expression. We try to link that to different biological processes, and we do find specific effects of specific protein sources. So we see changes when you compare the protein sources to soybean meal. That's what's presented here. So we see some common differences, but we see also specific differences between protein sources. And now we try to find out what is behind in terms of expression of genes and influencing different processes. We use the same kind of concept to see what early feeding could mean for piglets. This is an example on how we used caesarean-derived piglets to associate them with specific microbiota on the one hand and to feed them a specific diet on the other hand. So we did a study with piglets in which we did oral association with either a complex or a simple microbiota on the one hand, but this is more specific to show you the effect of diet. We fed these piglets diet with a different fat source, being either a combination of soya oil and palm oil, or we exchanged that in part for coconut oil, which is rich in medium chain fatty acids, which could have antimicrobial effects, or it has been shown that they have antimicrobial effects. And this graph depicts to what extent the intestinal microbiota was different as affected by the association treatments and the dietary treatments. And what you can clearly see that in piglets of 3 weeks of age, we were able to induce very specific intestinal microbiota profiles in these piglets, which were grown up remote from their mother. We also measured here the effects of these treatments on gene expression in the gut, and we linked gene expression to specific biological processes. And you see here in these comparisons that we had especially differences related to processes which could be attributed to activity and functioning of the immune system. So when we made these comparisons, especially for the diets, that we could see that all kinds of processes related to activation of the immune system were affected by these dietary treatments. So I show this as an example because we do not have data yet showing that similar effects could also be induced by dietary protein sources, but I'm pretty convinced that they will. And of course, we have to find out what this means in the longer term. So we plan to do some more research also to see what the effect is of early programming of the intestinal microbiota, of the intestinal immune system on later performance and health. And we have to learn a lot about that, I think, in the future. How can we identify the potential capacity of bioactivity of protein sources? This is some recent work we did on prediction of that by, again, using different protein sources which were subjected to trypsin digestion. And with bioinformatics, you can find out, using all kinds of databases which are in the public domain, the number of proteins which are present and found in these specific protein sources. So you can see that when you go very deep, you can identify in these protein sources at least 40 up till more than 700 different, chemically different proteins in these sources. And what you can subsequently do, and that's what we did an in silico digestion, because once you know the exact amino acid profile of these individual proteins, you know the specific cleavage sites of the main digestive enzymes, proteolytic enzymes. You can predict peptides arising from protein digestion, and you can relate the composition of these peptides to bioactivity, which are described in databases which are, again, can be found in the public domain. So actually, that's what we did. And in this way, you can describe the potential bioactivity of these protein sources, and that's what I try to indicate here. So here you see the total bioactivity identified, classified according to different classes of activity, and that's mentioned here. And you can see by the different patterns of the color that protein sources have different potential bioactivities. And the blue parts of this graph relate to ACE inhibition, that say this is related to effects on blood pressure, because the databases are mainly filled with informations on studies for human health domain. But in the long run, I'm pretty sure we are going to fill the database also with trials with farm animals and pigs. So we will create here specific activities which are important for our animals in relation to their health, for example. So this is, let's say, a first start to identify the potential bioactivity of protein sources. And right now, in the studies with the different protein systems we are doing, we try to link the in silico, the predicted bioactivity, with some measured in vivo bioactivity by evaluating the gene expression in the gut of piglets fed these protein sources for a considerable period of time. So we'll do a lot more work, I'm convinced, in the future in this area. One additional thing, and this is basically my final slide before the conclusions, is that we are feeding not pure proteins, we are feeding protein sources. That means it's quite often a combination of proteins, carbohydrates, and some other constituents. And that's something to realize, because if you feed, for example, and this is an example of wheat extracts, which do contain also very specific polysaccharides, and these polysaccharides could also induce specific functional effects. And that has been shown for the seaweed extracts, and when feeding specific seaweeds and sometimes also algae, that you can see that these carbohydrates do have specific effects also on gene expression in the gut, and that is indicated here. And even studies have been done feeding sows in the gestation phase and lactation phase with these functional properties, and they were able to show that this benefit also performance and health of the piglets during and after the weaning phase. So it's important not only, if you talk about protein sources, not only to consider the protein fraction present, but also to consider other constituents which could contribute to functionality of these protein-rich ingredients. So coming to the conclusions, I think we can be clear that we will use more and different protein sources in the long run, associated to the fact that we need more protein sources for animal feeds, and we need to be efficient in the use of all kinds of byproducts which arise from, for example, food and energy production. The strict nutritional value, I think we are quite far with that. We have all kinds of tables which describe fairly well nutritional value when it comes to the protein value. However, when it comes to the non-strict nutritional effects, the functional effects, especially relating to gut health and immune development, we know some of these effects are shown some examples today, but they are still poorly understood and to a very limited extent only applied in practical conditions. We have to make big step forwards in order to be able to translate this also to our diet formulation. But I think to some extent this will be future for us as well, and not only in the human nutrition domain. So in the long run, I would say we have to include these functional values as a more integral part of diet formulation. And with that, I thank you for your attention.